Hi, I am Dan Waitzberg. I am a physician, gastroenterologist, associate professor of Department of Gastroenterology at the University of São Paulo Medical School, and also scientific director of GANEP, Nutrição Humana, and the genetic sequencing company Bioma for Me. I am here to have the big pleasure to interview Professor Ilana Livyatan, who is a geneticist working at Hebrew University of Jerusalem Department of Genetics and also at the Department of Molecular Cell Biology at the Weizmann Institute of Science. Hello, Professor Ilana. How are you doing today?
Good. I'm doing fine. I just wanted to mention a correction in my associations. I was at Hebrew University, and I'm currently a postdoctoral fellow at Weizmann Institute.
Excellent. Very well-known Weizmann Institute all over the world. Ilana, you and your team have just published a fantastic study at Science where you have been studying the microbiome of more than 1,500 samples from 7 human tumor types. And using state-of-the-art technology of sequencing and computing, you have demonstrated the presence of bacteria in tumors, in intracellular localization, in both cancer and also immune cells. Now, this is absolutely new. We have before reports of colon cancer and breast cancer, but not such a multicentric scope as you have doing. And then the first question I would like you to expand is breast cancer raises between the others because it has much more bacterial species, much more bacterial load and richness. And also the phyla are completely different from the others. Would you have any specific explanation for this?
Okay, so that's really a wonderful question because that's one of the main findings that we did after a comprehensive search across 7 tumor types. We were able to view the higher diversity and richness that you just explained in breast cancer. So first of all, that's what the data shows us. We even took a lot of care to do a process called rarefaction, whereby we make sure that we look at the same number of samples and compare those to the other cancer types to really be sure that this is a valid finding. So why would that be? Well, we were surprised, as you just mentioned, generally to see bacteria in tumors. And the breast is actually, it's a question whether it's an external exposed tissue or an internal tissue. We weren't as surprised to see bacteria in, let's say, lung, because we breathe in and breathe out, so there would be an opportunity for bacteria to get in there. But it was surprising to see the bacteria in breast tissue. We do see that there's a similar profile of bacteria in the normal adjacent to tumor tissue. So the question arises what the source of the bacteria may be, and it could be an endogenous microbiome in breast tissue. We've seen reports of other articles preceding us that show a bona fide microbiome even in healthy normal breast tissue that's sometimes for breast reductions. So it could be an additional place to the gut and to the other exposed surfaces that we're used to seeing microbiome in. It could be another tissue that has endogenous microbiome. I can expand on another reason that might be more tumor related, and this is a hypothesis we've been having in the lab for a while, is that breast cancer is really a very heterogeneous disease. It is not one disease, even if it's in the same tissue area. There's several molecular phenotypes that have been characterized. There's different receptors localized to different breast cancers. And the treatments are very varied and wide. And we actually think of this disease as multiple diseases, which is why if we lump them into one basket, term breast cancer, we might actually be talking about several diseases. And that's why the numbers might be higher.
Yeah, indeed, you have shown that there are difference between those ER positive and ER negative results. But still concerning to breast cancer, as you said, there are some differences of species and phyla, even in each tumor type. You know, we have just published in OncoTarget a paper about it is an in silico paper, but we have studied the breast cancer tumor cells environment and also adjacent tissue, like exactly like you did in your studies. And we have seen that the metabolomic profile is completely different in relation to the carbon 1 cycle, meaning folic acid basically. And as I remember, you also found something related to folic acid in your study in breast cancer. So my question, and this is really a tricky question, is would the bacteria, the different bacteria from the natural adjacent tissue that you found in the tumors, they are there because the tumor cells make a different metabolic environment? First question. So the cancer cells, of course, makes a different metabolic environment, or and then the bacteria are attracted by this different metabolic environment, or totally different, the bacteria come, they made a different metabolic environment, and then the cancer may develop. What do you think?
Okay. Yeah, the classic chicken and egg question, what comes first? So that's an excellent question. It probably would be a bit of both, but I'll expand why I think that way. We've actually seen evidence and actually recently published a paper in Nature Communications in collaboration with Professor Bachrach, actually showing that bacteria can home from an external source, such as IV injection into mice, to tumors in the breast, or to a tumor model in mice in the breast. And that can be mediated even by receptors. So one way of bacteria reaching the tumor and coming after the tumor is already established can be by transient bacteremia circulation in the blood system and arriving at a type of tissue that is already aberrant and displaying different types of receptors from normal tissue, and perhaps also providing comfortable space for the bacteria to thrive in, maybe immunosuppressed environment, like you said, metabolically active in a certain way that is beneficial for the bacteria. Bacteria can do many different things. They can process many different energy sources, and so they are better adapted to these various metabolomic systems. On the other hand, there is a question of whether bacteria can cause a cancer to develop. The question is what stage you're going to be looking at. So a bacteria landing there and then causing a whole tumor to develop seems a little bit far-fetched, specifically for the breast tissue. But we have seen some evidence for it to be helping the tumor develop, progress, or proliferate. Imagine that once the bacteria arrive at the tumor, whether it's from normal adjacent tissue or from the blood system, they have unique metabolic activities that our body might not contain. We might not have the genes that are required for the tumor to thrive and survive. Excess need for iron or amino acid requirements that go up heavily in the tumor, and the bacteria could perhaps be providing that additional biochemical factory, if you want to put it that way. So we don't have a clear-cut answer. It could be a bit of both. In biology, lots of times we see a few options happening. But definitely the metabolic aspect of the relationship between the tumor cells and the bacteria that they harbor or that are within the microenvironment is going to be a particularly interesting issue to figure out in the coming years.
Thank you, Dr. Ilana. Also, in your fantastic paper, it's written that some bacteria maybe share some niche difference. For instance, Fusobacterium nucleatum is frequent in colorectal, breast, and also pancreatic tumors. You know, all of these three tissues, they share like adenomatosis, they have glands, you know, they more or less are similar in such a way. So any idea of what this has happened?
Right. So, right, the Fusobacterium being in colon cancer is a bit more obvious of how it might get there because we have it existing in our oral cavity. It's one of the bacteria that causes periodontal infections, and then we perhaps may swallow it, and perhaps that can reach the colon niche. The question arises, as you said, how can it get to breast tissue, to deeper pancreatic tissue? So perhaps from the gut, it can get to retrograde into the pancreas, but the breast tissue seems to be that it might be able to enter, like I said, the bloodstream either from the oral cavity or from some bleeding in the colon and create a transient bacteremia and get to the breast tissue. But again, even if it could reach various tissues in the body, the target tissue, if we'll call it that, needs to be the proper niche for this bacteria to thrive in. We know that since it's an anaerobe, but the colon is known as anaerobic. Pancreatic tissue also has some anaerobic niches. Probably deep inside the tumor, there's an anaerobic niche as well. So it could thrive there. We really have to delve deeper into the actual metabolic abilities of Fuso. It's actually becoming a more and more carcinogenic recognized bacteria for these various reasons. So I think it would be interesting particularly to study this bacteria. It's also been detected in metastases of these various cancers. So it's particularly interesting to follow this specific bacteria. And we indeed used it as a positive control in our study to detect it in colon cancer and in the other cancers.
Excellent. Very, very interesting. But one thing that called my attention in your study is the interaction between some metabolites and bacteria. Let us pick the case of bone tumors where degradation by hydroxyproline was found by bacteria. But you also state that other bone diseases may have elevated hydroxyproline levels. Okay, so my question is again this more or less the same topic. The presence of bacteria in bone tumor could be attributed to the higher availability of hydroxyproline, so they migrate for there, or do you think that normal bone also has bacteria?
Okay, yes, so it's a similar question, but again, we can use it to expand on some other ideas. Number one, we have to remember that the tumor environment is in kind of an aberrant structured environment. So the tumor is obviously a neoplasm of cell growth, but it also creates a new vasculature. It needs to pull in blood cells and needs to get the oxygen supply. So it does provide an opportunity, if we want to put it that way, for different invasions of elements into the tissue space. Now, we haven't tested, we'll say, healthy bone tissue and looked for bacteria in there in and of itself. That would be a good idea to establish whether there's just microbiome endogenous in that tissue. What we do hypothesize, though, is that once a tumor invades a tissue, it number one, creates this vasculature and enables different things that wouldn't be entering the tissue to now enter the tissue. So that could be one reason why we would see the bacteria appear in the tumor space and then see the hydroxyproline, where in normal vasculature wouldn't even reach there. And secondly, we have the opposition of the immune system that we have to remember. Normal tissues should have normal activity of immune cells. So if bacteria reached those locations, it should be cleaned cleaned up, even if there was a wound or something which would allow bacteria in, we should have a response, an immune response to clean that up. But we know that the tumor environment, the microenvironment of the tumor is very different from a normal environment. And there are many immune cells in that environment, but they're also sometimes exhibiting a suppressed activity. So that could also be the reason why we would see bacteria there benefiting or using the fact that that this environment is suppressed immune-wise.
Thank you. Another topic that is of makes a lot of interest here in Brazil for our clinical oncologists is about the response or not response of melanoma to immunosuppressive agents related to the studies in the United States, in Houston, and also in Harvard about this. And you also did a part of this in your study. You have compared responders and non-responders to these immune checkpoint inhibitors, and you have found something different. You have found that Gardnerella vaginalis in non-responders. I have to say that I was astonished. Can Gardnerella be responsible for the non-response? And then from where this Gardnerella shouldn't be there, from where did this Gardnerella came?
Okay, so yes, that's definitely a good place to ask these questions. So yes, we were interested in melanoma in response to therapy, and we had a unique opportunity to test the actual local microbiome in the tumor microenvironment. What previous studies have done before is look mostly at the gut microbiome and the state of the microbiome of the person, we'll say. Researchers such as Gajewski, Wargo, Zidvogel, and we've worked with Jen Wargo on this as well. And we had a unique opportunity to look at the tumor environment of the melanoma tumors specifically. And we did characterize the microbes within them, and we had a very good clinical metadata about the patients and their response to therapies, immunotherapies in particular. So the results are what we found. We obviously reported on what was found and not what we hoped to find. I think Gardnerella is an interesting finding. In some of our cohorts, it was very high, and in some of our cohorts, it wasn't as high. So it could be environmentally specific. It could be some people living in one place have the Gardnerella, and in another place, they might have a different microbe that takes that role. Finding on both sides of the spectrum, like you said, in response or non-response, I think it's part of the issue in the microbiome field of being able to sufficiently reproduce results. And also, we have to remember that these are two different niches. So one is found in the gut, and we found in the tumor microenvironment. That could explain a difference. And we still haven't ironed out exactly which environment is the major environment that's influencing immunotherapy. Is it the gut environment as the main body environment that influences all immune outcomes, or is it really the local immune environment of the tumor that influences the local effect of therapy on the specific primary or metastatic lesion? So these are really questions that your question led to that we have to iron out in the near future. And one of the ideas going forward is to match, to take from the same person, to take the tumor sample and then matched gut sample, and then iron these types of questions out. Like you said, where would Gardnerella come from? Well, if it's in the gut, it could come from the gut, going through the blood system. Maybe there's some leakiness there, permeability, etc. We do know that Gardnerella vaginalis, given its name, is associated with women's health. It is one of the bacteria found in vaginal infections, but we didn't see any bias toward women or men, any gender bias in our findings. So not quite sure that indicates where the source is coming from.
That's very interesting because, as you mentioned, studies before were relating gut microbiome to a lot of different, even lung, even melanoma, and also. So now we are seeing different ideas. So this brings me to the idea that we are just a vehicle for bacteria. So we are nothing. I mean, maybe we can think better than them, but the other thing around, they decide what to do. Well, um, I have to go back to another question. Um, you have in the discussion, it's written a very nice paragraph that says that it's not possible to establish causal role or just an infection. So I would like you to speculate how we can solve this question, because this question, as you mentioned, the egg or the chicken, is going around for the last, I mean, in microbiota, last 20 years at least. So no one has already solved that. I say, when I ask this to experts, they say, by the end of the day, it doesn't matter. Well, I'm not convinced that it doesn't matter. Because if it does matter, I can take my precautions. I don't know. I would like to sequence my microbiota. I would like to take extra care in food or exercise. What do you think? How can we solve this enigma?
Right, so that's an excellent question, because since we've really started studying the microbiome in all niches, the question is, how important is the microbiome really for our health? We can pick out specific places where it metabolizes for us, but on the whole, how critical is it to maintain a healthy microbiome, and what is that healthy microbiome? Where is it measured? Where are the issues that are important? And the causal role of them. So if we look at it from a cancer view, from tumor view, we know examples of cancers that we can attribute a causal role to a specific bacteria, like Helicobacter pylori, but it's causal via inflammation, right? The bacteria creates an inflammation, and then chronic inflammation lasting long enough by some hand waving can lead to tumor initiation. But actually, in my opinion, I'm just speculating here, I think that might not be the main picture that we'll see coming out of the microbiome field. I actually do think, like you mentioned, we are a niche for microbes. If you look at it from the microbe view, and the fact that we have tumors for whatever reason, genetic or exposure to some kind of UV radiation, etc., we can accumulate tumors, and then the bacteria find that there's no vacuum in the world. They find that, they reach that, and there's some interaction that happens there. So it might be creating a novel niche bacteria-wise for them to populate. So I would speculate that it's less of a causal role and more of an environmental role. The tumor is a very unique environment, and then it is a place where bacteria, some bacteria, not all, could take up shop and thrive. Now, that doesn't mean that they won't have an influence. It doesn't mean that they won't crosstalk with the immune system and perhaps suppress it a bit and allow the tumor to thrive in the background. It doesn't mean that they won't provide a metabolic activity that is beneficial, perhaps providing an essential amino acid that the tumor needs to thrive and grow. So I don't think that we can ignore them, and it does matter in one sense, but there is a question of at what point you want to start looking at them. Do you want to look at them before you ever have disease and maintain that healthy microbiome? Or do you want to look at them once there's a disease and use them as a diagnostic? And also that will also influence the treatment options, whether bacteria need to be treated along with the tumor or ignored, right? And just treating the tumor tissue. So these are all really big questions that we're going to try to answer in the next few years. But yes, the recognition that the microbiome is playing some kind of role, even if it's not causal, it's definitely influential, and we'll have to recognize the influences and treat them as such.
Excellent. And we are approaching the end of our beautiful interview, and I appreciate that very much. Your answers are very interesting. And this is a question that surely will interest our clinicians. Do you foresee in the future, at least, a potential use of probiotics or even prebiotics for patients with cancer?
So I do in some cases, yes, because of the element that you mentioned before of the influence of response to immunotherapy. by the microbiome, and we'll figure out if it's the gut or the local tumor microenvironment microbiome. We'll see which one is more important for that. It could be important to know which microbiome you need in order for immunotherapy to work effectively, and then strive for that microbiome. And it can be a probiotic, but I also study a bit of the gut microbiome, and I know that if you put a probiotic but you don't feed it with a prebiotic, it might not take. So I think it will have to be an awareness, a combination of a probiotic and a supporting prebiotic. It can also be general lifestyle. In other words, these microbes are in our environment. We don't necessarily have to take them in from the outside. It could be a specific diet that promotes a certain microbiome, and that can be beneficial. But I have to say that cancer is a pretty tough disease, and sometimes it's caused by severe genetic mutation and things like that. So it's not going to be the only possible treatment for cancer. We're not going to be able to leave the chemotherapies and the other therapies and say, let's just get our microbiome on track and then we'll never get sick or we'll never have to have a different kind of treatment. So we're going to have to learn a bit more about the level of influence the microbiome has for each type of cancer and for each type of therapy setting. And then I think we'll have a better idea of the amount of influence it has. Yes.
Thank you so much. Maybe some of our, the people who are seeing us would like to communicate with you. Would you like to share your email with them in order to further questions?
Sure, yes, that would be fun.
Okay, so I thank you so much. It was a fantastic opportunity. I learned a lot with you, Ilana. It's fantastic. Thank you so much. And I hope that you and Weizmann Institute continue to give us, to the world, such a beautiful insights in microbiome as you have done already. And I wish you luck in your future research. Thank you so much.
Thank you for the opportunity to speak to you and to get the word out to the medical community in Brazil.